You learned introduction to metabolism. You have also learned few things about the metabolism and its clinical significance, and about clinical biochemistry. Like this, you have learned some introductory parts of biochemistry. Coming to the biochemistry syllabus whole, our goal in biochemistry teaching is to make undergraduate students understand the scientific basis of the life processes at the molecular level. So, what is happening inside a cell, inside the mitochondria, what is happening outside the mitochondria, what is happening. So, like this, we will divide this life processes into different biochemical processes at molecular level. And another goal is to orient them towards the application of this knowledge acquired in solving clinical problems. Ultimately, this is a medical course, and you are supposed to solve the clinical problems. So patients will come with many of the symptoms, and you have to identify the symptoms properly, signs properly, and diagnose and solve the problem. So this basic biochemistry knowledge helps you to do this job of solving various clinical problems. So objectives. So we want you to learn few information to get the sufficient knowledge, and also to get some skills of various diagnostic methodologies, skills of various biochemical tests, skills of examination, skills of history taking, skills of communication. Like this, various skills we want you to learn. We also want you to learn integration, how to integrate this huge biochemical knowledge into the clinical significance, having a clinical significance, ultimately diagnosing a patient and treating them. So knowledge, we are going to discuss in detail about the various knowledge aspect of the biochemistry syllabus in detail. So few of these you have already learned, like molecular biology, introduction to metabolism. Like this, we are going to study in depth. We also want you to learn skills. At the end of the course, the student should be able to make use of conventional techniques or instruments to perform biochemical analysis relevant to clinical screening and diagnosis. And you should be able to analyze, and you should be able to interpret investigative data and demonstrate skills of solving scientific and clinical problems and decision making. These are the skills we want you to acquire during this 1 year first MBBS course of biochemistry. We also want you to know how to integrate the knowledge acquired in biochemistry. So this should help you to integrate molecular events with structure and function of the human body in health and diseases. So these are the important objectives of teaching learning in biochemistry for the next 1 year. So we hope, I really hope that you are already well aware of this word, that is CBME. Can anybody tell what is CBME?
Central Board of Medical Education.
Have you heard this CBME first? Are you hearing this word for the first time? Or have you heard this?
No, sir.
No.
Before you, sir, answer.
Okay, CBME is competency-based medical education. Competency-based medical education. Now the MCI, and now it is the National Medical Commission, it has come up with a new curriculum called competency-based medical education. So if you see the syllabus of the previous batches and yours, the syllabus is almost same, but the method of delivery, method of teaching learning is little different from them. Here we are going to teach you, and you are going to learn based on the competencies you have to be achieved. Competencies is nothing but it is a competency. You can see here. So this is a competency table given by the CBME syllabus of National Medical Commission. If you go to the website of National Medical Commission, NMC, so and you type for CBME, you will get three booklets, volume 1, 2, 3. So volume 1 includes all the first year subjects, anatomy, physiology, and biochemistry. And whole syllabus of anatomy, physiology, biochemistry has been divided into many competencies, and they have been described in this format. So if you want to see this competency table, how to understand this competency table, so the first column is the competency number. So you can see here PY 1.1. PY is the short form of a subject physiology. IM is another short form of subject called internal medicine. So, and next one is 1.1. This is the competency number. PY 1.1. That means competency 1.1 in physiology. What is that competency? Describe the structure and functions of a cell. So this is a competency you have to attain at the end of this teaching hour. So like this, almost all the syllabus of all the three subjects, anatomy, physiology, biochemistry, have been divided into competencies which have to be attained. Previously, what used to happen is the syllabus heading will be given as a topic for discussion, and the teacher would the teacher wanted to discuss whatever he knows about the subject and its clinical significance, and the theory class will be over. But nowadays, in the according to this competency based medical education, it is actually more of outcome based education. We have to make sure that you people have attained this competency. So after the class of this competency, you should be able to describe the structure and functions of a cell. So this competency you have to achieve. Now it is mainly outcome based, whether you have achieved it or not. So we are going to analyze using the suggested assessment method. So there are different assessment methods given by the CBME curriculum, and we are going to assess you that whether you have acquired this competency or not. So if you see the first column, this is nothing but the subject name with the number of competency. Next is what is that competency? That is the description of this competency. Next is domain. In which domain this competency belongs to. So there are, according to the NMC and medical education, there are 4 domains. K stands for knowledge, S stands for skill. So it is here. K stands for knowledge, S stands for skill, A stands for attitude, C stands for communication. So these are 4 domains of learning. And all the competences belong to either one of these domains. So that domain is it has been given in front of the competency description. So this is K means knowledge. You should have some knowledge about this. There is no skill required for this competency. But if you see the next competency, elicit, document and present a medical history that helps delineate the problems and solve the case. So this requires knowledge and also skill. So if you see this, the most basic level of learning is getting knowledge. The next level of learning is getting the skill. So next level of learning is you incorporate that learned knowledge into your day-to-day practices. That is called attitude. And the last stage of learning is whatever you have learned and you have adopted from this, you have to tell to others. Communication. So you have to inform the patients. You have to give education to the patients when you come in contact with the patients. Communication is not only with the patients, how to talk with your peers, how to talk with the teachers, how to talk with other seniors, like this. Communication is another domain of learning. So next is levels of competency. Levels of competency can be K. K means knows. The level is only knows. The person knows. And the next level of competence is knows how. It's a higher level of knowledge in which the person is able to discuss or analyze the things. Next level of competency is shows. Shows is it's a skill attribute. That is, the person is able to identify or demonstrate few of the steps of a procedure, steps of a history taking, steps of a examination, steps of various experimental techniques. All those come under shows. Usually this is a skill attribute. Then shows how. Shows how again is a skill attribute. In this, along with the ability to demonstrate, he should be able to interpret a complex procedure requiring thought, knowledge, and behavior. So this is show how level of competency. And the last level of competency is performs under supervision or independently. So at the end of the course, you should be able to perform few of the competencies independently. So for this, first you have to perform under supervision of the teachers. So that will take place for the next 1 year. And at the end of 1 year, there will be some practical examination where you have to perform it independently. So this is a mastery level of competence. So this is the last level of competence. So like this, various levels of competence have been described for each of the competencies. For example, describe the structure and function of a cell membrane. This is a knowledge domain. You have to know about this. There is no skill required for this. And it is a know-how level. You should be able to know this. You should be able to describe this also. That is know-how level. Then next column is core. Core, the answer may be yes or no, Y or N. Core means, what do you mean by core? Here, few of the glossary has been given. Core means it is a competency that is necessary in order to complete the requirement of the subjects. So if you want to if you want to pass the biochemistry subject, you have to acquire this competency. Yes, core, because it is a core competency. So these core competencies have to be acquired by all the students to pass the examination. So that is the meaning of core. What is the meaning of non-core? Non-core competency means it is desirable. It is good to know. If you know something, it is good, but it is not mandatory. So all those are given as non-core, N, non-core competencies. Next is suggested teaching learning method. So what type of teaching learning method you want to use for this? So mainly this is a knowledge based. So we want to use lectures, small group discussions like this. So the teaching learning methods have also been suggested by the NMC for all the competencies. And how do you want to assess whether the person has acquired this competency or not? Those methods have also been suggested by the MCI or the now National Medical Council. So it is written examination or viva. So like this, and along with vertical integration, horizontal integration. So more focus has been given on this integration part, especially vertical and horizontal integration in the medical curriculum in this competency-based medical education. So usually what used to happen is in the preclinical subjects, the syllabus, usually it is of non-clinical or preclinical department level. That means there is no much interaction with the patients, no much interaction with the clinicians. Only preclinical teachers used to teach this. But now, according to CBME, we have to integrate more. Horizontal integration is integration with other departments in the preclinical subjects. So preclinical subjects are 3: anatomy, physiology, biochemistry. So biochemistry department can horizontally integrate with anatomy and physiology only. But if the biochemistry department wants to integrate with surgery, medicine, like this final year subjects, that comes under vertical integration. like this, what type of integration should be done for each competencies. All this have been in detail, the NMC guidelines have given. So those who are interested to learn about this competency based medical education can go to the NMC website, National Medical Commission website, and if you put the keyword CBME, you will get 3 volumes of books. In that volume, first includes the preclinical syllabus, that is anatomy, physiology, biochemistry. So our biochemistry department, biochemistry department has been allotted 250 hours of teaching to the first MBBS students. So these 250 hours includes theory and practical. Theory is 160 hours. Theory includes lectures, small group discussions, and case-based learnings. So the biochemistry department has got all the infrastructure required for all of these types of teaching learning activities. Small group teaching means a small group of 20 to 25 will be there, whereas in lecture all 150 students will be delivered a lecture in the lecture theater, whereas small group teaching takes place in different small class settings where 25 to 30 students will be accommodated, and so that more personalized discussion can be done in small group teaching. And also 40 hours have been allotted for case-based learning. So more importance has been given for first MBBS students to learn about the occurrence of various signs and symptoms, diagnosis, and management. All those included in this case-based learning. Next, practical includes almost 70 hours. And then one more type of teaching learning method introduced by the CBM is SDL, that is self-directed learning. Here, there is no much role of the teacher. Here, the students are active learners. The students are given some topic. They have to search in the textbooks. They have to go to library and search various textbooks, reference books, internet, and they have to collect the knowledge, and they have to come and discuss with the moderator. So that is self-directed learning. 20 hours have been allotted to this self-directed learning. And for the first time in the CBME, early clinical exposure has been incorporated. What do you mean by this is, usually in the second year, the MBBS students used to go to the hospital and they start seeing the patients, how they present with what are the major history, how the clinical symptoms occur, what are the signs, all these things. The students used to learn in the second phase, but not in the first phase. Actually, there was absolutely no patient interaction in the first phase. But according to the subject experts, now the NMC has decided to introduce early clinical exposures, ECE, in which the first MBBS students only are, the clinical material will be brought to the first MBBS students. So first MBBS students are exposed to the patients from the first year onwards itself. So this is for 30 hours. We have to do 12 hours for clinical skills and 18 hours for basic science correlation. So now the syllabus has become almost the topics which we would like to discuss remain same, but the mode of teaching learning is little different, in which small group teaching has been given more importance and self-directed learning has been given more importance. And for the first time, EC has been started. So like this, biochemistry has been allotted 250 hours in that. So we have divided these according to the guidelines of the National Medical Commission. So this is a sample competencies table in the biochemistry. So the topic is basic biochemistry. Time allotted for this is 1 hour. Number of competencies to be achieved is 1. Number of procedures that require certification, nil. Only the practicals which the students are supposed to do independently, they require certification. So if you see the first competency, that is BI 1.1, BI is biochemistry 1.1. This is the first competency of biochemistry. So this is describe the molecular and functional organization of a cell and its subcellular components. So this is just a knowledge domain. So there is no skill required for this. So this is knowledge domain. And what is the level of competency? This is know-how level. The first level is knows. That means you just know some information. Knows how means you know the information and you know how to describe it. That is the knows how. So we want you to attain know-how level of competency for this. Then whether it is core or not, why? It is yes. That means all students must learn this. All students must learn to describe molecular and functional organization of cell and subcellular components. So what is the suggested teaching learning method for this competency? It is mainly the lecture and small group discussion. The suggested assessment method is written examination or vivas. So like this, and we can integrate with the horizontal integration with the physiology department for this first competency. So like this is an example of one competency table of the basic biochemistry. So coming to the knowledge of biochemistry, knowledge component of biochemistry. So we will discuss various topics of biochemistry syllabus now. So I'll just briefly describe you the topics, and the in detail discussion of all these along with the clinical importance is very essential. So whatever you learn in this MBBS course, that knowledge is first part, that is the basic knowledge. And the next what you should look for is how to apply this in clinical field, how the how it is related to the production of diseases, manifestation of diseases, how it helps in the diagnosis of diseases, how it can be used for the treatment of diseases. So all these matters you have to think for all these basic subjects, including anatomy, physiology, biochemistry. You will learn, these are called as basic subjects. That means which gives you fundamental knowledge. But also you should be able to learn. You should be able to ask whenever whenever the teacher finishes this basic subject, you can ask what are these applications in the clinical field, how it can be used for the diagnosis and management of patients. So that component has to be kept in mind. So the first topic we discuss in the first MBBS is cell and its subcellular components. I'm sure all of you are aware of the different types of cells: prokaryotic, eukaryotic, animal, plant cells, its features, its differences. But here we concentrate mainly on human cells. So we are not concentrating on any other organism. We are not concentrating on plants. We are not concentrating on any other animals. It's only humans. So you have to know about the structure of human cells and its subcellular components. That is the first part. Next important part is chemistry of various biomolecules. So since the topic is, since the subject is biochemistry, we have discussed in the first class what is biochemistry. It is the chemistry of biological or biological systems. So chemistry of various biomolecules you have to learn first. So biomolecules includes carbohydrates, lipids, proteins, nucleic acids. So what are the, how do you define carbohydrates? What are its, how they look like? What are their molecular structures? Their properties of each of the different carbohydrates, their classification, and ultimately clinical significance of each type of carbohydrate you are going to learn. Similarly, the same thing with the lipids. Lipids are water insoluble substances. Examples: oils and fats. So you are going to study what are lipids, what are their chemical and physical properties, what are the how do you classify different types of lipids, and each of the lipids how they are important for the medical field, their clinical significance you are going to learn. So whatever you learn along with the basic topic, you should learn some clinical significance of that substance, which will be asked in the exams also. If you add this clinical significance of each of the parameters, you'll get more, you will fetch more marks. Again, the same thing, chemistry of proteins, how proteins are made up of. Proteins are made up of amino acids. So they are the polymers of amino acids. So what are the different types of amino acids? what are their properties, how do you classify them, what are their functions, what are their clinical significance of each of the amino acids and each of the proteins present in the human body. We are going to learn in this chemistry of biomolecules topics. Another is chemistry of nucleic acids, that is DNA, RNA. DNA, RNA, what are the basic units of DNA, RNA, and what are its significance, everything you learn in this chemistry part. So the next important and very big part of biochemistry is this one: digestion, absorption, and metabolism of various biomolecules and their integration, how they are interrelated. So here you are going to learn about carbohydrates, lipids, proteins, and nucleic acids. How the different types of carbohydrates are digested, how the different types of carbohydrates are absorbed, how the different types of carbohydrates are metabolized, and what is their significance. Here you are going to learn in depth about the metabolic part of various biomolecules, including carbohydrates, lipids, proteins, nucleic acids. So here you will get one more important topic, that is inborn errors of metabolism. So if you are aware of few biochemical pathways like glycolysis or Krebs cycle, I'm sure all of you are very well aware of the glycolytic pathway, where one molecule of glucose will be broken down to produce pyruvate. And this pyruvate enters the Krebs cycle as acetyl-CoA. And at the end of Krebs cycle, you will get NADH, FADH2 molecules, which will again generate energy in the electron transport chain. So this is how the basic metabolism look like. So if you take the example of glycolysis, it involves various steps catalyzed by various different enzymes. These different enzymes have different properties, and the word inborn errors of metabolism arises from this, where there is congenital deficiency of some enzyme leading to halt of that particular biochemical reaction, so that it will not occur, it will not continue further, and the substances which are before this enzyme will get accumulated. The substrates will get accumulated because the enzyme is deficient, enzyme is abnormal. So you will learn enormous number of inborn errors of metabolism of carbohydrates, inborn error of metabolism of lipids, inborn errors of metabolism of proteins and nucleic acids. So this disease part, how these diseases appear and how this can manifest in various patients, and what is the biochemical or molecular basis for this, what is the exact defect you are going to learn in this. So the digestion, absorption, and metabolism, this is a major part of the biochemistry syllabus, which constitutes around 50%. So if you take this chemistry and metabolism, it constitutes around 70% of, or 60 to 70% of whole biochemistry syllabus. So the whole one semester you will dedicate for learning their digestion, absorption, metabolism, and their clinical significance, along with the diseases associated with the pathways. Next is metabolism of few other substances of biochemical importance, like heme and hemoglobin we are going to learn. Can anybody tell what is heme? Does anyone know heme?
Iron.
Blood. Iron, sir.
Okay.
Iron blood.
Okay, all of you are aware of the word hemoglobin. Hemoglobin is the protein present in blood which carries blood gases, that is oxygen and carbon dioxide. All of you are aware of hemoglobin. In this hemoglobin, there are two parts. One is globin chain. Globin is protein, and another is non-organic part. Non-organic or inorganic part of hemoglobin is called as heme, which includes one uh, one molecule of iron. So heme contains iron, and heme along with globin forms hemoglobin. So the organic part of hemoglobin is globin, and the inorganic part of hemoglobin is heme. So you will learn how these heme and hemoglobin are synthesized, because the metabolism involves both the things, anabolism and catabolism. Anabolism is how this molecule is synthesized in the body, and catabolism is how this molecule is broken down in the body. So similarly, for all these carbohydrates, lipids, proteins, some carbohydrates are generated inside the body, that is anabolism. Some carbohydrates are broken down, that is catabolism. So altogether, you will learn about the metabolism. So metabolism of heme and hemoglobin and its associated disorders, very important topic for the first year examination and also for your clinical practice. One more topic is plasma proteins. You know what is plasma? Where is it present?
The fluid part of the blood without the cellular components.
Yes, very good. So if you see the blood contains water part and the cells. If you separate these cells, whatever remains is plasma protein. Whatever remains is plasma. So this plasma contains all types of biomolecules. And there are some proteins in the plasma which have various significances, various clinical significances. So you will learn in detail about what are the different plasma proteins, what are their functions. For example, one important plasma protein, can anybody name? It is albumin. So albumin is present in plasma. That is a major plasma protein. So like this, we will classify the plasma proteins. You will learn what are the functions of each of the proteins and what are the clinical significance of each of the plasma proteins. That is again one part of biochemistry syllabus. The next part, section 2 of biochemistry syllabus, contains various biologically important substances like enzymes. Enzymes, one class has been taken on the enzymes. Enzymes are biocatalysts. They are the catalysts of the living world. So they increase the rate of reaction. So they are of huge implications, and absence or deficiency of one single enzyme can alter the whole pathway, can stop whole biochemical process that may lead to huge clinical implications. So enzymes, how they act, what are the factors regulating enzyme activity, how enzyme activity can be regulated, how enzymes can be inhibited, how this can be used for clinical diagnosis and therapeutics. You will learn in this topic. Next is vitamins. Vitamins, again, very important topic, where vitamins can be divided into fat soluble and water soluble. You will learn about fat soluble vitamins of A, D, E, K, and water soluble vitamins of B complex and vitamin C. There are some important minerals in the human body, like macro minerals and micro minerals. You will learn in detail about the minerals and their clinical significance. What is the function of each of the minerals and what are the diseases associated with those minerals deficiencies? You will learn. So one more important chapter is principles of nutrition. So here you learn what are the nutritional aspect of carbohydrates, proteins, lipids, how much a person should take, what is the BMR, basal metabolic rate, and what are the diseases associated with less nutrition and more nutrition, called as malnutrition. So malnutrition includes both overnutrition and hyponutrition. So this is nutritional deficiency disorders like kwashiorkor and marasmus you will learn, and nutritional disorders like obesity also you will learn in this nutrition part. One more important section is molecular biology, where you will learn about the metabolism of nucleic acids, replication, that is duplication of DNA molecules, transcription, conversion of DNA molecule to mRNA, and translation, production of proteins from mRNA, and regulation of gene expressions, how different genes are expressed in the humans and how this can be regulated and how this can be used for disease prevention. All this you will learn in depth about the molecular biology. One more important section is clinical biochemistry. So this has a direct application with the clinical field. That is, clinical field means this is actually related to the patients. related to the patient diagnosis, patient's treatment, patient's monitoring, etc. So some part of biochemistry is directly related with the patients. So that we call it as clinical biochemistry. Here you have to know principles of various laboratory investigations and instrumentation analysis and interpretation of laboratory data. Suppose if you take the example of diabetes mellitus, can you tell me what is the problem in diabetes mellitus? What is the basic defect? Can anybody tell me what is the basic defect in diabetes mellitus? Have you heard of diabetes mellitus?
It's
Efficiency.
decrease in glucose level.
What water
High
is
blood
going?
sugar.
Yes, very good.
Blood sugar
So
level.
the major feature of diabetes mellitus is hyperglycemia or increased blood sugar. So how do you know the blood sugar is increased? For that, you have to undergo
Presence in urine.
investigation. Some laboratory tests will be there. For example, fasting blood sugar, that is a laboratory test. If you give your blood sample, the laboratory will take that blood and it will analyze using different machines, using different instrumentations, how much of glucose is present in this blood sample. So that is given as blood sugar level or blood glucose level. So there is some normal range of blood sugar level. If that glucose is the concentration of glucose is above this range, we call it as increased glucose level or hyperglycemia, which is a characteristic feature of diabetes mellitus. So if you want to diagnose a person as diabetes mellitus, the person has to undergo laboratory investigation, that is blood test. What blood test? Estimation of sugar, estimation of glucose in the blood. So that is a laboratory investigation. So like this, for different types of diseases, there are battery of laboratory investigations. There are numerous tests should be done in order to come to a conclusion. In order to come to a conclusion of what is the diagnosis and how should we manage this patient. So for that purpose, clinical biochemistry is required, where you will learn about different laboratory investigations and how they are measured and how to interpret the laboratory data. So laboratory will give you result that your blood sugar level is 200 milligram per deciliter. Now you should know how to interpret this, whether this level is normal or not. So if this level is high, can we call it as diabetes or not? Like this, you should also know to interpret the laboratory data. So this part includes water electrolyte balance, acid-base balance, different organ function tests, how the liver is functioning. For that, there are some battery of investigations. We call it as liver function tests. Similarly, how kidney is functioning, renal function test, how thyroid gland is functioning, thyroid function test, how stomach is functioning, gastric function test. Like this, for different organs, different biochemical tests have been devised, so which will give you a fair idea about how they are functioning. So all these come under clinical biochemistry. Here also you will learn about different types of hormones and their mechanism of action, their significance and their related disorders. You will also learn about biochemistry of cancer, HIV/AIDS in the clinical biochemistry. So clinical biochemistry is very important for your clinical practice, and a sound knowledge of basic biochemistry and clinical biochemistry will help you excel in the next few years. Coming to the practicals, I will not go in depth about the practicals. Whenever you come here in the campus, we will discuss in detail about different types of practicals, how to take these practicals and how to do these practicals on your own. So mainly the practicals can be divided into qualitative experiments and quantitative experiments. Qualitative experiments means it is, as the name indicates, it is about the quality. It is about what are the types of sugars present in the solution, what are the different properties of these sugar solutions. Like this, you will learn, that is qualitative experiments. Quantitative experiments means you will learn how to estimate blood sugar level. So if you learn that comes under quantitative experiments. And there are some demonstrations which you need not perform. You just have to look for how